Today, we are very fortunate to have Dr. Monica Uddin from USF, University of South Florida, down in Tampa. Dr. Uddin started off her career, as any good scientist does, by studying primate genetics. So we have lots of connections, and I know many of us here have an interest in understanding primate genetics. But that passion quickly developed into trying to some really interesting research and trying to understand how aspects of our environment or our experiences get under the skin to influence our genomes to ultimately impact our health, well-being, and survival. Dr. Uddin did her undergraduate at Stanford, and then she went to her PhD at NYU, where she worked with Cliff Jolly, studying different types of Papillonins. Then she did a postdoc at Wayne State, became a professor there at Wayne State, moved over to University of Illinois at Urbana-Champaign, and is now at USF. I'm really excited to have you here, and I hope you all really enjoyed reading the papers that she sent around to us. And today she's going to tell us about her work entitled Imprints of Social Adversity and Traumatic Stress on the Leukocyte Methylome. So please join me in welcoming Dr. Uddin.
Thank you, Noah. Thanks everyone for the invite and for the opportunity to speak with you today. As Noah mentioned, I'm going to be giving you an overview of some of the more recent work that's been conducted in my lab. And I'm very glad that you had hopefully a chance to read some of the background papers that I sent, because a lot of what I'll be talking about today will be more of a bird's eye view rather than down in the weeds, but I'm happy to address any questions you may have later if I don't cover things thoroughly enough. Okay, bear with me while I see how my slides advance. Aha, okay. Here's the outline of what I'll be covering today. What I focus on is the impact of stress broadly defined, and I have a particular focus in my work on post-traumatic stress disorder and trauma exposure. So I'll give a brief overview of what that actually is, so we all have the same vocabulary moving forward. I also want to briefly touch on why study epigenetics in relation to PTSD. And then I'll be focusing on two specific studies that are ongoing in one way or another. The first is an examination of stress, trauma, and social adversity in a cohort that is no longer recruiting participants, but we have many samples that we're continuing to refer to for our ongoing work. It's called the Detroit Neighborhood Health Study. And the second is an ongoing study in Rwanda examining the intergenerational impact of exposure to the Rwandan Genocide against ethnic Tutsi. And I hope you had a chance to read the New York Times article giving a bit of historical background about that event, because as you can imagine, in a 40-minute talk, I don't have enough time to give that horrific event the due diligence it needs so that people understand it. But I do hope that you had a chance to read it. If not, please go back and read it after the talk, and what I say might make more sense. Finally, I'll touch really briefly on the importance of genomic diversity in genomic analyses moving forward, and how the H3Africa Consortium is contributing to this effort, and end with some concluding thoughts. Okay, so PTSD. We're hearing a lot about that these days, especially in relation to the COVID-19 pandemic. The most current definition of PTSD in the psychiatric manual known as the DSM-5 refers to a combination of symptoms that occurs after exposure to a traumatic event. And these symptoms have four main domains. According to this psychiatric Bible, they involve re-experiencing, avoidance, negative alterations in cognition and mood, and alterations in arousal. And these four symptom domains can't have been present prior to the exposure to a trauma, and they need to persist for at least a month to be eligible for a diagnosis of PTSD. I just want to pause and define trauma as well, because we all have an intuitive sense of what trauma means. Obviously, it means something that's an extreme kind of stressor that induces a psychological and physical state that is not often encountered in the everyday, but there is a unique characteristic that is required for traumatic event exposure in relation to PTSD, and that is an aspect of life threat. So if you either directly experience an event in which you think your life is being threatened, or you witness somebody else's event in which their life is being threatened. That would qualify as a trauma. It's not the same as suddenly losing your job or going through a divorce. Those are very stressful, but they don't actually qualify as trauma. What's interesting to me is the fact that a majority of adults are exposed to what we call a potentially traumatic event, a PTE, because not everybody will necessarily appraise an event as potentially threatening. So we call them PTEs. We will all be exposed to one or more PTEs during our lifetime by the time we're an adult, but only a minority of us go on to actually develop full-blown PTSD. So there's a disjunct between trauma exposure and the actual diagnostic category of PTSD. So as Noah mentioned, I'm now at USF. I'm in a College of Public Health. I did some training after Wayne State at University of Michigan's College of Public Health. So I think it's really important to contextualize complex disorders like PTSD in relation to the epidemiology of the disorder. And so we know that mental health conditions are actually the leading cause of disability adjusted life years. That's DALYs for short, worldwide, and they account for 37% of healthy life years lost from non-communicable diseases. So DALYs refer to years of life lost, not just due to death, but also disability. And so they're a more comprehensive measure of the impact of a particular disorder. And so here's a ranked ordered list of different mental disorders in terms of DALYs. PTSD is not at the top, but we know the lifetime PTSD prevalence across the globe is near 13%. It's not insignificant. And we know that it has a high cost in terms of DALYs. Relatedly, we know that PTSD, as well as many other disorders on this list, have significant physical medical comorbidities. So PTSD is known to co-occur frequently with diabetes, with cardiovascular disease, and other disorders. So though we like to think of PTSD as a disorder that is of the brain, it actually has a physiological component too. Okay. When we consider the prevalence of trauma exposure across the globe, these are estimates from the World Mental Health Survey, we can see that there is quite a bit of variation. On average, the exposure to trauma is approximately 70% globally, but you can see that, for example, in the US, 82.7% of the population has been exposed to one or more traumas, versus in China, the estimate is a little bit lower at 52.5%. Now, this gets at trauma exposure, not PTSD. So how does this translate to PTSD? Well, I mentioned before that there's a disjunct between trauma exposure and actual PTSD development. This graph shows us the 12-month prevalence of PTSD in select countries around the world. And you can remember from the prior slide, the United States had a prevalence of about 82.7% trauma exposure, but you could see down here at the bottom, the prevalence of 12-month PTSD is relatively, I wouldn't say low, but it's lower than Northern Ireland. Northern Ireland has the highest, whereas its prevalence of trauma exposure was only 60.6%. So there are characteristics that go beyond prevalence of trauma exposure that kind of need to help us understand how you get from trauma exposure to getting PTSD. And that's what I'll be talking about a little bit today. There's heterogeneity in some of these factors. Here in the United States, what I'm showing is the prevalence of traumatic event types by race ethnicity. As you'll soon see, the Detroit Neighborhood Health Study and of course the study in Rwanda are focused on African ancestry individuals. Here in the United States, we know that the prevalence to overall trauma is actually lower among African Americans than among other ethnic groups. But when we look at distinct traumatic event types, there are differences, some of which are shown here. This slide is showing you black-white differences, which is indicated by the asterisks, in indicating significant differences in traumatic type event exposure. So you can see that participation in organized violence, sexual violence, and other traumatic event types is significantly higher among individuals of African American ancestry versus white individuals. And all this to say that these differences in traumatic event types can sometimes translate into differences in PTSD prevalence. Not all traumas are equally created, if you will. Some traumas have what are called a higher conditional risk of PTSD So for example, if you're in a car accident, you are less likely to get PTSD than if you are sexually assaulted. So some traumas are inherently more likely to precipitate PTSD. What I'm showing here are lifetime prevalences of PTSD by race ethnicity. And what we know, not from this particular study, but a related study, is that even when we do consider traumatic event types, African Americans are at higher conditional risk of PTSD. So it's not just that they have higher exposure to intimate partner violence or interpersonal violence. It is, even when we control for trauma characteristics, there's something else that needs to be considered to help us understand why individuals of that background in this country are at increased risk. These might be things like discrimination, exposure to environmental toxins. All of these have been hypothesized as potential reasons mediating this apparently enhanced vulnerability to PTSD. What do we know broadly about what causes this disorder? Well, we know from twin and family studies that both genes and the environment contribute to PTSD, with genes explaining about 35% of the variance in risk and the remaining 65 or so being explained by the environment. Of course, this doesn't tell us which genes or which environmental factors make that contribution. It just gives us a snapshot that both are important. So this is where epigenetics comes in. So as you've probably learned in one or more of your courses by now, epigenetic mechanisms refer to those biologic mechanisms that can alter or regulate gene expression without modifying the underlying DNA sequence. And so there's a range of epigenetic mechanisms being shown on the slide: histone modifications, microRNAs, DNA methylation. Many of these regulate DNA accessibility and thereby help to sort of determine the extent to which the surrounding genes are turned on or off. We know now, although we didn't 30 years ago, that these mechanisms are very often influenced by age, environmental exposures, and disease. So they're these sort of malleable or plastic molecular mechanisms that can help us understand the impact of the environment on our biology. Today, I'll be focusing specifically on DNA methylation, which is an epigenetic mechanism that's relatively stable compared to some of the others I just mentioned. So this refers to the addition of a methyl group to the 5' position of cytosine, most frequently when cytosine is paired on the same strand of DNA next to guanine. So these are called CpG sites. These are stable modifications that are typically propagated through subsequent cellular replications. They are tissue specific. So for example, we can't necessarily say that if a particular CpG site or a gene region with many CpG sites has high methylation in one tissue like blood, it will also have high methylation in another tissue. And so today I'll be talking largely about DNA methylation measures that were obtained in leukocyte, white blood cells. So just keep that in mind, because we can't necessarily generalize to other tissues. Why epigenetics in relation to PTSD? Well, PTSD is actually unusual among psychiatric disorders because it requires an environmental exposure in the definition of the disorder. So if you aren't exposed to a trauma, you're not even eligible for a PTSD diagnosis. And so figuring out among those exposed to trauma who actually gets PTSD and who doesn't is a key question, and epigenetic mechanisms may help to explain these observed differences. When we're looking broadly across the genome and we see which sort of genes are impacted on an epigenetic level by something like PTSD, it'll help us understand the biological processes involved and potentially help us develop targeted therapies down the line. Okay, for the first study example I'm going to be talking about today, the Detroit Neighborhood Health Study, I just want to give you a bit of background on this particular project. So this is a project that was launched back in 2007, and it went on till 2013 in terms of data collection. It's a probability sample of households in Detroit, and one randomly selected adult per household was sampled within that framework. Our baseline sample had just over 1,500 adult participants recruited as part of this cohort. And the study included a 40-minute structured telephone interview in which people were asked about their exposure to trauma, PTSD symptoms, depression symptoms, anxiety symptoms, but also questions about their neighborhood, about their chronic health conditions, about their drug use, and related questions. So in the second wave, we also were able to include some questions about childhood maltreatment that weren't asked of everybody. So one unique feature about the study is that survey participants were geocoded and linked to residential neighborhood based on their zip codes. And this has enabled some really interesting work to be done based on neighborhood level variables that I think you'll be hearing about from one of my colleagues, Chantal Martin, in another seminar in the series. Importantly for the work that we've done in my group, a subsample of participants provided a biologic specimen at each wave. And so this was a set of consenting participants who allowed us to come into their home, take blood, or if they didn't want to give blood, we took a blood spot. One year we gave the option also of doing saliva so that we could potentially link biomarkers of stress to what we were seeing in their structured telephone interviews. So this is a snapshot of the DNHS study participants at baseline. The red dots each indicate one participant. In the blue there is Hamtramck, which is a separate city within the city of Detroit, sort of separately regulated, and the island is Belle Isle, so there are no residents there. So this just goes to show that we had fairly decent coverage across the entire city. For our currently funded R01, which draws on the existing data from the parent study, the overall goal that we're examining is the extent to which DNA methylation in glucocorticoid receptor regulatory network genes, GRRN genes, might be a mechanism linking social adversity and stress-related psychopathology in urban-dwelling African Americans. So the DNHS is overwhelmingly an African American cohort. What I'm showing here is sort of the study design of each wave among those participants who actually gave a biospecimen. So we had 5 waves of the study. Our baseline wave, as I already mentioned, had about 1,500 participants and about 400 gave a venipuncture specimen. And we followed up each wave with as many people as we could. We didn't collect biospecimens in wave 3. We did collect blood spots, but not venipuncture. And then we followed up as many as we could till wave 4, and then a subset of people in wave 5 who had children. And so this is sort of the data frame that we're referring to for the study that I had you read. There have been many different publications from the DNHS. So what I'm presenting today is just one study that kind of gets at the question of which social adversity factors in combination with methylation variation might shape risk for PTSD. So why are we looking at GRRN genes? Well, as you probably know, the hypothalamic pituitary adrenal axis gets activated when we experience stress to distribute cortisol through the blood, which then goes on to regulate gene expression in virtually every cell of the body. And since we're working with blood, we wanted to make sure that we could access methylation measures in a pathway that was stress relevant, but also detectable in a peripheral tissue. So we turned to this particular pathway. The GRRN genes all help in one way or another to regulate the glucocorticoid receptor encoded by the gene NR3C1, which you may have heard of. And these have been implicated in PTSD and other stress-related mental disorders in a whole host of previous studies. So this is our working model. We hypothesized that external stressors such as low socioeconomic position, stressful life events, psychosocial stressors might act in concert with variation in DNA methylation in these GRRN genes to influence risk for psychopathology. And for this particular study, we're looking at post-traumatic stress symptom severity. We didn't look explicitly at depression or generalized anxiety symptom severity. So the research questions we had are, what are the significant factors among these various predictors, social adversity, prior psychopathology, and GRRN DNA methylation that are associated with prospective risk of PTSD? So first is to identify the significant factors. And then using those factors, how well can we predict the prospective risk of PTSD? And this is work that was led by my postdoc, Dr. Agazwani, whose picture is down on the right. The paper that I gave you is Impress, and I gave you the uncorrected proof, but a corrected proof should be coming very, very soon. What are the data that we assayed? So the methylation data were captured using the EPIC-B chip by Illumina, which measures DNA methylation at 850,000 CpG sites in the human genome. So DNA methylation using this method is measured on a continuous basis that can range from 0 to 1. If a CpG site has a low methylation value, like this one, it will have a value closer to 0. But if you look here, this is a different CpG site for individual number 7. There's a much higher beta value, we call them, which indicates higher methylation levels at this particular CpG site for this individual. You can see there's variation here, right? Individual 0 has a lower methylation level at this CpG site than this individual. And these are the measures that we used as part of our data matrix for our machine learning models. Now, I will say that 850,000 CpG sites sounds like a lot. We only focused in on the GRRN CpG sites, of which there were about 2,000 or so, because we had a prior hypothesis about their role in shaping risk for stress. So this is just a snapshot of the data that we drew on for our actual analysis. So we drew on DNA methylation data from waves 1 and waves 2. And we had social adversity and psychopathology data from waves 1, 2, and 3. Remember, I mentioned we didn't do venipuncture draws in wave 3, so we didn't have methylation data from this wave. But we used data from waves 1, 2, and 3 to predict PTSS at wave 4. That was our outcome, if you will. We weren't able to do anything with wave 5 because of its limited nature. So our data involved 148 unique participants, overwhelmingly African American. 62 of these participants had repeat measures of DNA methylation. So for our long data set, we had 210 rows. We applied a machine learning approach to sort of understanding the relationship among these important features to predicting PTSS. And machine learning is a little bit different than some of the other approaches you may have heard of in epidemiology when we try and estimate risk. Machine learning, there are different approaches that you can see described here, but it doesn't use an explicit programming if-then kind of approach to unpacking the relationship between the predictors and the outcomes. It learns and it trains itself from the given data to find patterns that are predictive of the outcome. So you might be familiar with methods like WGCNA that uses unsupervised learning to cluster genes that are maybe similarly methylated or similarly expressed. We use supervised learning because our data actually had labels, and we used a lot of regression type approaches because our data was continuous rather than categorical in the outcome we were examining. And reinforcement learning, to my knowledge, hasn't been used that much in genomics, but I'm sure that's coming down the line. The whole field of machine learning has enjoyed an explosion in the past 5-10 years and is increasingly being applied in health-related research. So a key step in the machine learning approach was to pre-process our data to identify the important features among the many, many thousands of features that we had at our disposal to focus in on the ones that were most predictive of our outcome. So without going into the details, because I'm assuming that you've read the paper, we had a long data set and we had a wide data set. So the wide data set were for those individuals that had repeat measures of methylation, and we had one row represented unique individuals, whereas the long data set stacked samples, which may have come from the same individual. And so the long data set had fewer features than the wide data set because the wide data set essentially had CpG sites from both waves 1 and 2. But in both cases, we tested for association with PTSS in this feature selection step. And then we selected the top 150 features from each data set. And In the case of the long data set, we had 79 CpGs and 71 phenotypes. In the case of the wide data set, we had more CpGs and phenotypes. But in common to both, we had 95 features, 44 CpGs and 51 phenotypes. And so with this sort of reduced set of features from both the long and the wide data set, we then instituted machine learning prediction of PTSS. So we did this for this set of features in the long data set and for this set of features in the wide data set. And here is a summary of the 95 significant features, which are both CpGs and phenotypes that were common in both the long and the wide analysis. Of course, I don't expect you to be able to read this, but hopefully you were able to scrutinize it a little bit on paper when you were reading the paper. What you can see is a rank ordered list with the features of most importance up at the top. And so we're showing the importance of the feature in negative log scale. So closer to 0 means more important. And you can see up at the top that wave 3 post-traumatic stress score was the most important feature in machine learning. And then this was followed by wave 2 PTSS score. In terms of the social adversities that we were looking at, loneliness was the topmost ranked social adversity feature. I believe it was 28th on this list, followed by perceived discrimination and financial problems. So you'll see that there are many CpGs listed, but interspersed with them are phenotypes. And you can see that loneliness and the other two that I mentioned are on this y-axis as well. So if we hone in on the top 10 significant features that were common in both long and wide analyses, you can see that there's a dominance of psychopathology symptoms in terms of their importance in their predictive abilities. And it's not limited to PTSS. We have lifetime depression symptom severity here too. But we do have some CpGs that are popping up in the top 10 as being important. And these are, again, GRRN CpGs, AKT1, and one you may have heard of a lot in relation to psychopathology, which is FKBP5. And so that's been studied quite a bit in relation to PTSD, childhood maltreatment, depression. I could go on. And it's popping up here in our analyses as well. So if we break down the top 10 features in the long and the wide analyses separately, what you can see is there are some differences between the top 10 most important features. But again, up at the very top, I want to draw your attention to the fact that the most important feature in both data sets is wave 3 PTSS. And this importance score here shows that it is responsible for 89% of the predictive power in the long data set and 88% in the wide data set. So of all the 150 features, this topmost feature is eating up almost all of the importance because the remaining 149 features are going to sum to 1 in addition when added to the wave 3 PTSS score. So the dominant feature in all of these analyses was prior psychopathology in terms of post-traumatic stress psychopathology. We tested 7 different machine learning models. The primary paper reported on 3 across all the different versions of the study that we did. The best performing model was gradient boost. I have to love peer review because we had a really great suggestion from our reviewer asking us what the models would look like if we took out wave 3 PTSS. What would happen? So what I'm showing here as a result of this request is our original analysis, which I just mentioned. If we take out wave 3 PTSS, the topmost feature is wave 2 PTSS. It's more important in the long data set than the wide data set, but it's still the top ranked feature in both. And without both wave 3 and wave 2 PTSS, what's the topmost feature? Well, wave 3 lifetime PTSD. So not a continuous measure of symptoms, but a categorical measure of whether or not somebody had lifetime PTSD at wave 3. So what we're seeing is even without the topmost feature here, there's always some sort of weighting of prior psychopathology in terms of importance in predicting future psychopathology. Okay, I mentioned that you can't directly relate what you see in the periphery to other tissues, including the brain. But one of our aims in this award is to try and start to understand the relevance of stress in real time in CNS relevant tissues. And so just really quickly, what we are doing, this is more of a pilot study that we're still working on, is we're looking at human microglia and trying to figure out whether when we expose them to the stress hormone cortisol or its synthetic analog dexamethasone, do we see altered gene expression or DNA methylation in our genes of interest? The reason we're doing microglia is that it's a follow-on study from some earlier work in the DNHS that demonstrated that there was elevated levels of monocytes in men with PTSD. And we also know from rodent studies that rats exposed to repeated social defeat seem to have trafficking of peripheral monocytes into the brain that become microglia. So there's sort of a biological relevance between the periphery and the central nervous system by looking at these cell types. So this is really quick, our experimental design. We take proliferating microglia. We treat them with either dexamethasone or cortisol for 3 days. And then we do a 3-day washout to see if there might be persistent effects. We extract DNA and RNA both at 3 days and at 6 days. And then we look at the gene expression and eventually methylation of our genes of interest. And what we've done so far is to look at specifically the GRN genes in our cell lines. And we have found that there's an upregulation of EGR1, which is a transcription factor known to bind to genes like the glucocorticoid receptor, as well as FKBP5, which you saw in our earlier results, and downregulation of another gene, KRT17, which is part of the GRN pathway as well. We're starting to look at methylation. We're still in our early days with that. And we are also trying to verify whether the cell line we're working with is actually African ancestry, because it would be nice to know whether our peripheral findings, which are overwhelmingly from African Americans, are mirrored at all by a cell line of ancestry relevance. Okay, so this is just to pause and tell you about the overall summary from the first study that I just went over. So we tried to use machine learning models to tease out the important features among our rich data set of psychopathology, social adversity, and genomic features to identify risk factors that predicted prospective risk of PTSS with high accuracy. We saw that prior PTSS ranked highest in predicting the prospective risk, accounting for more than 88% of the prediction. But we also saw that social adversity factors, some of which have been explicitly hypothesized to explain the higher conditional risk of PTSD among African Americans, including perceived discrimination. We saw that factors like that also are implicated in PTSS risk. We are trying to build upon this work using data sets from the Psychiatric Genomics Consortium to kind of test whether these models generalize. So that's the future work in this area. Okay. So now I'm going to switch gears a little bit and talk about the second study, which is the study of the Rwandan Genocide. Now, I was telling Noah earlier that I am guessing that many people on this call were not necessarily even alive when the Rwandan Genocide occurred. And so it is such a notable historic event of high significance that I cannot do justice to with a single slide. And I really hope that you had a chance to read the article to understand the historical significance of what went on. This is the orientation I can give you in the short time that I have. So the Rwandan Genocide against the ethnic Tutsi is also known as the 100 Days of Killing, where in 1994, upwards of 800,000 people were killed, 1/7 of the population, largely ethnic Tutsi who were targeted by Hutu militias, and almost 2 million people in the country were accused of participation. So not only is there the trauma of people being essentially hunted, but there is also the trauma of the perpetrators as well. The census in 2005 showed that 1 in 4 children were orphans due to this event. Clarice Musana-Baganwa, who's a trainee on the grant that we currently have examining the impact of the genocide, she's a genocide survivor herself, recently conducted a meta-analysis and identified that The prevalence of PTSD in post-genocide Rwanda sits at about 25%, but among genocide survivors specifically, that prevalence is much higher, 37%. So the impact not only to the survivors, but also to the offspring of the survivors and eventually the grandchildren of the survivors is something that really needs to be considered, because we're having, unfortunately, more and more opportunities to understand the impact of trauma across generations. So this particular study is funded by the Human Hereditary and Health in Africa mechanism, which is a mechanism that is designed to promote genomic research in Africa, funded by the NIH, the Wellcome Trust, and the African Academy of Sciences. And our particular study has a goal of characterizing the intergenerational impact of genocide exposure on PTSD in women survivors of the Rwandan genocide and their offspring. And a component of this that is actually even more important than the research component is scientific capacity building. So this grant is as devoted to scientific capacity building as it is to the research question that's being posed, and that's how the mechanism was designed. So this is just a quick snapshot of the project design. As I mentioned, scientific capacity building is a big component that's kind of covered here. Our research has been delayed by COVID, as you can imagine, but involves participant recruitment and epigenomic analyses, as well as psychological analyses. And we're also doing community outreach to make sure that people are aware of the work that we're doing on the ground. So science is inherently collaborative. International science on a topic like this is especially collaborative. This is a snapshot of our study team. It doesn't get at everybody who's on the ground actually interviewing study participants, but we have 4 PIs, we have 3 co-Is, and we have 4 trainees, all of whom you can see here. And what we're trying to do is to understand the epigenetic relationship between exposure to the Rwandan genocide and subsequent psychological symptoms. So this was the best visual that I could find that related sort of our study design in terms of a complex disease outcome. It's not a mental health outcome, it's type 2 diabetes, but the idea is the same. What we're looking at is women who were in Rwanda and exposed to the genocide while they were pregnant and their resulting offspring. And we're comparing them to ethnically matched Tutsi women who were pregnant during the genocide as well, but outside of Rwanda, and their resulting offspring. Because the idea is that exposure to an extreme stressor like genocide may very well impact the epigenetics, not just of the mother, but also of the developing baby. That's a very plastic time in development, whereas you probably know epigenetic mechanisms are extremely labile to begin with. Sorry, that's my daughter. So this is sort of the idea behind this study as well. We're building on a pilot study that Dr. Mutesa published some years ago now that identified that women exposed to the genocide compared to women not exposed to the genocide had much higher PTSD and depression symptoms, and the same was true of their resulting children. So he did a study of 100 participants, 25 in each quadrant, and also collected biospecimens from these individuals. This sort of gave us the impetus to do our currently funded work where we're building on that and recruiting new participants. He also looked at methylation in the glucocorticoid receptor, finding differences between mothers who were exposed versus not exposed to the genocide and the children who were exposed versus not exposed to the genocide. He also found that cortisol levels in the exposed group here were lower compared to the ethnically matched individuals who were not exposed. And this might be a little counterintuitive, but cortisol has actually been found oftentimes to be lower among people with PTSD, rather than higher, as you might expect, like in depression. In our current study, we're delayed with our recruitment, as I mentioned, but we are trying to not just recruit the mothers who were pregnant during the genocide and the child born from this pregnancy, but also the younger sibling. So that's a new addition compared to the pilot study. And it's sort of a proxy measure for whether or not there might be persistent epigenetic effects. This is a snapshot of one of the community engagement efforts where the PI, Leon Mutesa, went out to try and inform the community who might be interested in participating in this study why we were doing it, engage interest. This is our main research objectives for the research side of this study. So we were interested in determining genome scale DNA methylation levels in participants exposed versus unexposed to the genocide using an EWAS approach, and then identify epigenetic differences associated with intergenerational transmission of trauma and PTSD. And so again, we use the EPIC-B chip. And what we're doing here, because we're still recruiting new participants, is we're looking at the pilot samples that were tested for NR3C1 that I showed you a few slides ago. So we were fortunate enough to still have 70 samples from those 100 participants, and we used them to test with the EPIC chip. So as you can imagine, having 70 samples on a chip that assays 850,000 CpG sites introduces a multiple hypothesis testing problem. So we needed to reduce the search space for our analysis. So what we did was we selected the top 5% of probes based on variance because they would have a higher likelihood of showing differences. And then we used a package called McC to identify differentially methylated regions, specifically promoter regions, between children who were exposed and unexposed to the genocide, and then mothers who were exposed versus unexposed to the genocide. We found 16 significant DMRs in the children and 24 significant DMRs in the mothers. So this is just really quickly showing you what those 24 significant DMRs are in the mothers. So this is a comparison again of just mothers exposed versus unexposed to the genocide. So within one generation. And many of these are genes known to be important in developmental processes. And here we have 16 significant DMR genes in children exposed versus unexposed to the genocide. Again, many of these are also related to developmental processes. Shared between these two lists are these 6 genes. And again, these are differentially methylated regions. So these are looking at multiple CpGs in a region. But if we're trying to get a sense of what the differences in methylation are, we can drill down and look at individual CpG sites. So here's an example of the BCL6 co-repressor, and we can see that among children exposed to the genocide compared to their non-exposed counterparts, there's an increase in methylation. And there's a similar direction of effect for the mothers who are exposed to the genocide compared to the mothers who were pregnant at the same time but not exposed. So increased methylation in the mothers as well. This again is all being done in blood. Now, of course, we're always interested in what's going on in the brain. So there are databases that you can consult to infer their relationship with central nervous system tissues. One database is called ImageCpG. And of the 5 genes shared, or at least 6 genes shared with the mothers and the children, there are 3 that have a high correlation, a high known correlation between brain and blood within individuals sampled for this database. So this suggests potentially that at these specific CpGs in these specific genes, what we see in the blood could possibly be indicative of what's going on in the brain, but we would need to do more follow-up work for that. Okay, so just to summarize this study, we see how abnormal HPA axis activity is related to PTSD, and that epigenetic modifications may contribute to the transmission of traumatic stress from mother to offspring. That's based on the pilot study that Dr. Mutesa did. In our ongoing work, we actually identified several common DMRs in the children and the mothers. And even though we don't know if this is going to hold up in the participants we're actively recruiting for now, we do know that based on the pilot study, there is a clear biologic embedding of traumatic stress that has endured more than 20 years after the genocide. Okay, I know I'm running out of time. I just want to give a plug to the need to understand genomic diversity and how the H3Africa Consortium is contributing to that. Really quickly, there is a disparity in genome-wide association study data in the publicly available GWAS catalog. This has gotten better over time, but still is not great. There's a preponderance of individuals of European ancestry in the database as of 2016. This is true even for more updated estimates. On the left, we see ancestry category distribution of studies in the GWAS catalog. And if we consider it at the individual level, it's even worse. So we don't really know the extent of diversity in populations outside of the European continent. And why is this a problem? It's a problem because there may be ancestry-related differences in genes and other genomic measures associated with PTSD or whatever complex disorder. So what I'm showing here is a Manhattan plot where anything above the red line is a significant variant associated with PTSD. On the left, you can see that in European ancestry individuals, there are several hits in chromosome 6. On the right, you can see there are a few hits in African ancestry individuals, but they're on chromosome 13. Now, without sampling the African ancestry individuals, we wouldn't know that. And it would be a little dangerous to generalize that what's on the left applies to the population on the right. If we think about things in more immediate terms, specifically COVID-19, this is a small study that came out in the Journal of the American Medical Association back in September, looking at variation in nasal gene expression of the transmembrane serine protease TMPRSS2. And what this gene does is it mediates viral entry of COVID-19. And you can see that among self-identified Black individuals, expression of this gene is quite a bit higher than in any other ethnic group. And so without understanding how ancestry contributes to potential variation and risk for disorders, we're kind of going to be flying blind in terms of managing disease risk. It was known before this study that variation in this gene often left African American men at higher risk for prostate cancer, but now it appears that there are other liabilities to this variation as well for people of this background with other disorders. So H3Africa is trying to address this gap by promoting research across Africa for projects that are examining the relationship between genes and the environment for a multitude of different disorders. There are 3 explicit mental health related projects, including the one I just went over. There are many others related to diabetes, blindness, stroke, things like that. I encourage you to go to the website of this consortium because it's a really rich resource and it's in its second and final round of funding. But even once it's done, I think it'll be a resource that you can draw on for decades to come. Okay, so I'm sorry that I went a little bit over. Just a couple of concluding thoughts. I want to revisit the idea that population-based studies do confirm a higher conditional risk of PTSD among African Americans, even when controlling for trauma-related characteristics like traumatic event type. This suggests that additional biological and social factors need to be identified that mediate this risk. And that's what the first study I went over was trying to address, identifying social factors that might otherwise get missed that do seem to influence prospective risk of PTSD. Across both studies that I went over, I think it's pretty clear that we are in our infancy when it comes to identifying ancestry relevant factors that prospectively do this, both within and between generations. But it's really through these consortium efforts with everybody being as collaborative and collective as possible that our understanding of the genetic influences on the epigenetic signatures of complex disease, including PTSD, will improve. And so with that, I'd just like to thank you for your attention. I'd like to thank members of my lab, both recent and current, who are shown here. Not all of them are shown. Sorry about that. Haven't been together to take a picture in some time. I want to thank my current grant support. And again, thank you for your attention.
Thank you so much, Monica. Lots of claps, thumbs up. Suhail really appreciated your explanation of machine learning too, which is great. Does anyone have any questions?
I just want to say thank you for your research. This is important and it's really interesting, especially the difference between European and African ancestry people, the difference in the predicting genes for PTSD. And thank you for the time. So, yeah.
Thank you for listening. I know I went on for a while.
Nolan, I want to point out that we had a talk last fall by Alicia Martin, where she talked about a lot of those differences. Well, you see, if you look through our website, you can see one of the recordings where it goes into a little bit more detail about what Dr. Uddin just told us. Anne?
I have a question. Or do you want me to go ahead? Okay. Fascinating talk. Thank you so much. One question that comes to mind is in relation to the Tutsi genocide, are you, and I guess I think about this in because of the previous part of your talk about the Detroit project, are you measuring levels of PTSD in the individuals in Rwanda and comparing people that fall into the group that have a PTSD and those who, for whatever reason, don't, even though they went through the same event?
The short answer is yes, we are doing that. In the participants we're currently recruiting, people who were not exposed to the genocide do still have PTSD symptoms because they're going to have been exposed to other things themselves, but the symptoms and the rates of diagnoses are higher in the exposed group.
And do you see a sex difference?
We have not done that analysis yet. We're relatively underpowered with the pilot data that I showed you the results from. In the paper that Dr. Mutesa published in 2014, they did do a sex analysis and didn't find any differences by sex. We're going to have to be very careful about how we do it with the participants we are recruiting, because an N of 50 is still relatively small. So if we split that by sex, and we do expect there are going to be some sex differences, but we may be underpowered to detect anything at all. So one thing I didn't mention is this H3Africa consortium has as part of its mandate that any study that's collecting biospecimens, which all of them are, has to give it to a centralized biobank in Africa so that other investigators, first African primarily, and then later on people from other places in the world, can draw on them for other studies. So that will help increase power for some of the questions that you're asking. And we've done some harmonization with other groups to figure out what mental health phenotypes actually exist across more than one study. And so that could help us get at improving power to ask some of the sex questions that you're interested in.
Oh, cool. Thank you.
I had a quick question. So I'm wondering, I know you've done the research in Africa, but are there any studies that you're familiar with that kind of have the same kind of research and insight in regards to like American chattel slavery and the epigenetic effects on that, and possibly Reconstruction, civil rights, things like that, like kind of the same equivalency of the Rwandan genocide, but kind of here in America?
The short answer is I can't think of any off the top of my head that deal with those historical events that you just mentioned. That being said, I know the National Longitudinal Study of Adolescent Health, AdHealth, which is a population-based study of originally youth, and now they're like in their 40s, is collecting epigenetic measures that are population representative across all of the US. And some of their surveys in their later years do have questions about PTSD. And I know that they're collecting epigenetics as well. So that should be coming in the future in terms of like a nationally representative database of epigenetic measures. I doubt that they have questions about the history, though, that you're talking about. There are studies that have looked at other genocides, like the Holocaust. So
Right.
Rachel Yehuda's work is something you might be familiar with. And I am a co-investigator looking at the impact of historical trauma in Native Americans, specifically Alaskans. And I'm not driving that work, but that work is still ongoing. So there's some work going on in relation to other historical events in the US. that I'm aware
All right. Thank you. Great talk, though. And go blue. I'm a big Michigan fan. So.
of. Okay.
I will please join me in thanking Dr. Uddin for a wonderful and captivating presentation today.